Hello friends, welcome to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. Today we are going to talk about cancers. This is a series of lectures where we will talk about cancer in the details in the upcoming lectures also. So today we are going to talk about introduction of the cancers and the characteristic feature of the cancer cells. So what we are going to cover in this whole series of lectures, we will talk about basics of cancers. Then we will talk about the characteristic feature of cancer cells. So these two portions we are going to finish today. Then we will talk about the molecular basis of cancers, where we will talk about the etiological features and different etiological properties, like how cancer is going to cause. So we will have chemical, physical and biological carcinogenic agents. So then we will talk about the genes which is involved in the cancers. So there we will talk about proto-oncogenes and oncogenes and the tumor suppressor genes. So these two are the important genes where normally mutation in these two genes ultimately going to cause cancers. So we will talk about these two genes in the details. Then apoptosis and tumor marker. Tumor marker is one of the important sections where many questions comes into the different competitive exams. Then we will talk about the cancer therapy which is available to us till date, and the different strategy for the cancer vaccine development, and that we will talk about. So these are the different portions which we are going to cover in this series of lectures. So first we will talk about basics of cancers. Actually cancer is nothing, it is an uncontrolled growth of abnormal cells anywhere in the body. So two specific word we are going to use it, that is uncontrolled growth and for abnormal cells. First, cell, normally in our cells we are going to have a regular cell division, means whenever our cells divides, our cells normally divide in a regulated fashions. So cells, whenever body needs, then division will be there, cell will divide, and that will be in the regulated fashions. It will divide up to the moment till we want. But in case of cancers, cells are going to be unobedient. They are not going to follow the normal instructions, normal signaling, normal instructions given for the regulation. So that normal control mechanism is going to be failed. So cells becomes abnormal, and because of that abnormal, cells are going to divide uncontrolled fashions. So the cell division will be in uncontrolled way. This cancer is normally, it is a genetic disorders where normally gene is going to be modified, gene is going to be mutated. Sometimes this cancer is also genetically transferred from one generation to the next generations, but more than 90 to 95% cases, yeah, maximum cases, cancers are caused because of the avoidable reasons, that is environmental factors which can be avoided. Only 5 to 10% cases cancers are going to be caused because of genetic, means that will be hereditary, which is going to be transferred from one generation to the other generations. This abnormal cells which is going to be unobedient are called as cancer cells. It is also called as malignant cells, or it is called as tumor cells. There are many factors which is going to cause cancers, which initiate cancers, which is normally increasing the risk of cancers. Those are tobacco use, because it contains many carcinogenic agents which is going to cause cancers. Some of the infections, these infections may be from bacteria, from virus, from fungus, from parasites. So different kind of infections can lead to cancers. Some radiations, different. In case of radiations, we are having basically two types of radiations, ionizing and non-ionizing radiations. Any radiations which will have as much as energy, so the radiations which have maximum energy is going to cause more damages, and the radiation which is having less energy is going to cause less damage. So radiations is going to cause the damage. Then lack of physical activity. If we are going to live more sedentary lifespan, not doing too much of physical activity, not going to do too much of exercise, in that case cells active, means cells normally responsiveness towards any signaling component is going to reduce, and same thing is going to happen in case of obesity also. So whenever we are going to do live the sedentary lifespans, not going to do exercise, not going to do physical activity. In that case, cells responsiveness is going to reduce. And last is environmental pollutants. These pollutants or any of the factor which we have seen earlier, either tobacco or infection or radiations, ultimately going to affect our DNA. And once DNA will be modified, DNA will be mutated, it is going to lead to cancers. So in any case, mutation is the compulsory component for the going to cause cancer. So mutation is necessary. Approximately 5 to 10% of the cancers are entirely hereditary. I have already told you maximum most of the cases it will be environmental. Only 5 to 10% cases it is going to cause because of the genetic. Before going to see further details, we should understand some basic terms. First term is neoplasm. Neoplasm means any abnormal growth of a new cells. So whenever a cancer is going to happen, it is going to form a tumor, granuloma, deposition of normally many cells, abnormally cells are growing, and it will give you a swelling type of features. That is called as neoplasm. So new growth of a cells, abnormal cells are called as neoplasm. This is of two types. Either it will we are going to have benign, which normally going to be curable. It will be fixed at one location. It is not going to detach from the locations. So it is called as benign. Second one is malignant, which is normally cancerous. This is the malignant, is the true really cancerous. This is actual cancer cells, which is going to spread throughout the body. Next term is tumor. The word tumor normally refers to a mass, a granuloma, a deposition of tissue. This is general terms that refers to benign, generally harmless or the curable, or malignant, which is cancerous, which is detaching from the original place and spread throughout the body. Benign, already I have told you, these are normally non-malignant, it is curable, it is harmless, it is non-cancerous tumors. Whereas malignants are cancerous and it will be detached from one location, it is normally non-treatable. This malignant tumors normally not going to respond any kind of treatments. So once tumor becomes malignant, it is very difficult to treat it, very difficult to manage it. And last term is cancers. So cancer is another word normally we are going to use for malignancy, malignant cases. So I have told you malignant are the true cancer cells. So normal cancer cells, the normal healthy cells, in our case normal body health cells, normal healthy cells, is have going to differentiate, divide, in ultimately die in a sequential fashion. Whenever body requires, it will differentiate. Whenever body needs, it will divide. Whenever body needs, it is going to die. But cancer cells is going to lose the control over the differentiation, over the division, over the death. So that is called as cancer cells. So cancer cells does not differentiate in a normal fashions, does not divide in a normal regulated fashions, does not die according to the normal fashion. So its cancer cell is going to be immortal also. If you see the picture here, this is a normal cells. So normal cell divide, one cell divide into two cells, this cell divide again, and again it is going to divide, and ultimately cell is going to die, suicide, through apoptosis. Whereas in case of cancer cells, cell divides in irregular fashions, in uncontrolled fashions, and cells of irregular shape. So in this case you can see almost all the cells will have regular shape and size, whereas in case of cancer cells their shape and size is going to be modified, going to be changed. So we have seen cancers are two types, benign and malignant. The basic difference is that benign cancer cells are encapsulated, they are covered with a capsulated, there will have a capsule covering. Whereas malignant tumors, malignant cells are non-capsulated, there will no be any kind of covering, no capsulation, no encapsulated will not be there. Benign tumors does not invade to the local tissue, so it does not affect the normal local tissue, it will be restricted at a specific locations. Whereas malignant tumors invade local tissue and affect the local cells also. Benign tumors are going to be fixed at located at only one location. It does not detach from the original place and it will be restricted to the only one locations. Whereas malignant tissue are going to be metastasis. Metastasis means it will be detached from the original place and move through circulation through blood to the other parts of the body. So it will be spreaded throughout the body parts. And then malignant tumors are also destructed. It is going to for destructions. There are different other terms we are using according to the cancers, which type of tissue is going to be affected or what is the origin of the cancers. If the origin of cancer is from the epithelial cells, we are going to call it carcinoma. If the origin is epithelial cells. If origin of cancer is from the connective tissue, we are going to call it sarcoma. If origin of cancer is from the lymph or lymph nodes, we are going to call it lymphoma. If origin is from blood cells or blood, we are going to call it leukemia. So different type of terms we are using in case of cancers. Here you can see the picture of benign and the malignant. In case of benign, it will be encapsulated and it will be restricted at one location. These benign tumors are not going to be spreaded. It will not going to remove from the original place. Whereas in case of malignant tumor, we don't have encapsulated. That capsule is going to be broken and cells are going to detach from the original place. It will be spreaded through the blood throughout the body. So cells are cancerous and can spread to the other tissue and organs. Now the benign tumors and malignant tumors can be differentiated on different ways. So the tissue benign and malignant can be differentiated on the basis of these specific terms, which we can use a mnemonics like derm in map, DERM, derm, in MAP. These are the specific mnemonics we can use for the differentiation between the benign and malignant tumor. D means differentiation. Differentiation is a terms which we are using how the cells which is modified, it looks similar to the normal cells, original cells from the original cells. How much similarity is there? So that is called as differentiation. Encapsulations means covering. Rate of growth or loss of contact. Normally, because rate of growth is very fast and these cells are going to lose the contact inhibitions. Contact inhibition means when two cells comes to close to each other, if their cells periphery comes to the close to each other, they will stop their growth. So normally our normal cells are going to grow in a single layer on the substratum, whereas because of loss of contact inhibitions, cancer cells grow in multiple layers, multi-layers. So that is because of loss of contact inhibitions. Metastasis, when it is detaching from the original place and move to the other locations, and this metastasis is caused because of loss of anchoring nature. Anchoring nature means normal cells needs a kind of platform, a kind of substratum, a kind of surface to grow. So if we are having a normal cells, that requires a surface area for growth, for their development, for their division. But cancer cells does not needs this support system, so they can grow without the support system also. So the anchoring power, anchor, does not required for the cancer cells. Invasions, it is going to affect the nearby tissue or not affect, going to invade the nearby tissue, that is called as invasions. Nuclear to cytoplasmic ratio. Normally in a normal cells, we are having less space covered by the nuclear DNA, and whereas cytoplasm is going to cover maximum space. Normally 1:4 ratio is, or 1:4 to 1:6 ratio is for nuclear cytoplasmic ratio. Mitotic rate, the cell division rate. Anaplasia is loss of morphological features. If the morphology of the cells is going to be changed, going to be lost, going to be changed, modified, that is called as anaplasia. And polymorphic means different cells will have different kind of morphology. Angiogenesis, formation of new blood vessels, and escape from the apoptosis. normally will not have normal cell division. So these are the basic characteristic feature of a normal cancer cells. So if we will see the differentiation, in case of malignant cells, differentiation will be high, means the normal cells and the modified cells will be completely different. They will not be very similar. Encapsulations, benign malignant tumors does not have any kind of capsules. Just now we have seen in the pictures, they have not any kind of encapsulation. They are capsule free, so that they can spread from one location to the other locations. Rate of growth, the benign tumors, malignant tumors is having very high rate of growth, very fast they are going to divide, they are going to replication rate will be very fast. Metastasis, that is going to detach from the one place and move to the other locations, that is going to occur in case of malignancy. Invasions, obviously in malignant tumors invades local tissue. Nuclear to cytoplasmic ratio, normally I have told you the ratio is 1:4 to 1:6, but in case of malignancy, because number of chromosome replication is very fast, so chromosome replicates and we are having very high amount of chromosomes, so the nuclear size is also increases, and in that ratio cytoplasmic ratio is going to decrease. So nuclear to cytoplasmic ratio is 1:1. Mitotic rate very high because it is replicate very fast. Anaplasia, normal cells morphology is going to be lost, that is going to happen in case of metastasis. Pleomorphic, different cells will have different kind of morphology. Why? Because in case of cancers, the cytoskeleton's rearrangement, cytoskeleton's normal features is going to be lost, and normally the cytoskeleton cells, normally fibers is required to keep cells in a proper shape. So because cytoskeleton is going to be modified, so cells will have irregular shape, different type of shapes, and sometimes cells will have irregular plasma membrane also. It will not be in the regular fashions. Different cells will have different sizes, different surface area, plus angiogenesis. For cancer growth we require new blood vessels formation, that is called as angiogenesis. That is also going to happen in case of malignant cells, and escape from the apoptosis. So normally normal healthy cells going to die with the proper programmed cell death, that is called apoptosis. Cancer cells are immortal, they are not going to die in a normal fashions, so no apoptosis is going to happen. When we are going to see benign tumors, so almost benign tumors having a normal, it looks like a normal cells. So differentiation, no, there is no differentiation, it will be almost same. Encapsulated, it will be rate of growth will be very less. Metastasis, it is not going to happen in case of benign. It does not invade through the local tissue. The nuclear to cytoplasmic ratio is according to the normal cells only, 1:4 or 1:6. Mitotic rate normal, no anaplasia, no pleomorphic nature, no angiogenesis, no escape from the apoptosis. So benign tumor behaves normally as a normal tissue. So this is normally non-cancerous, non-tumorous. Whereas like mole you can see, warts you can see, these are benign tumors. Mole of our body, warts that is present in our body, that is benign tumor. Whereas malignant tumors, these are the real cancer cells. Now we are going to see the cancer cells characteristic feature, whatever we are going to see. So cancer cells will have irregular shapes. Why it will have irregular shapes? Because the cytoskeleton of cancer cells is going to be changed, and once cytoskeleton will be changed, so the cytoskeleton is normally responsible for the proper maintenance of cell shape. So say when cytoskeleton will be not in the regular manner, it will be irregular, so obviously cells will have irregular shape. Large or darker nucleus, I have already told you, because chromosome in case of cancer cells, chromosome is going to replicated very fast, so more chromosome number will be there, more chromosome amount will be there, and when chromosome number will be more, so obviously chromosome will be dark color or intense color. Because of that one only, cytoplasm nuclear to cytoplasm ratio is going to affected, so it is 1:1. In case of cancers, normally normal cells divide whenever they get a signal, but the cancer cells divides without a proper signal also, without a proper growth factors also. So because of that one only they are having very high rate of cell divisions and they proliferate uncontrolled fashions. So when the normal cells grow in a normal tissue mediums, they grow and divide at a rate similar to that of cancer cells. When the normal cell proliferate and cover the bottom of the culture dish, whenever we are going to culture in the laboratory, so normal cells cover the surface of the culture dish. Their growth rates decreases when they come to the close to each other. So if two cells come to the close to each other, their growth speed will be reduced. That is called as contact inhibition. So normal cell grow in a monolayer and they, because of their contact inhibitions, whereas cancer cell does not have contact inhibition, so they grow in a multi-layer fashions. So they no contact inhibition will be there. Immature cells, the normal cells are going to differentiate, going to divide and then mature and perform the normal functions, whereas cancer cells are, because they replicate very fast, so they don't go for maturation. So almost all the tumor cells, cancer cells are immature cells, so they don't perform the normal function of the tissue. Loss of communications, so cancer cells does not respond to the normal signaling processes, so they lost the communication systems. High angiogenesis, because cancer cells when it divides, so new tissue requires more growth factors, growth components, nutrients, oxygen supply. So because of that one, new blood vessel needs to be formed. So in case of cancer cells, angiogenesis rate is going to increase. So high angiogenesis process will be there. Other than this one, requirement of oxygen will be high, because oxygen provides them energy, so the more oxygen is required by the cancer cells. Not only oxygen, because oxygen without cannot give energy, so it requires glucose also. So more requirement of oxygen for energy production, as well as more requirement of glucose. Main source of energy is glucose only, so more glucose production will be there, because it is going to metabolize more. So it is going to convert it into, and if oxygen will be not sufficient, then more lactic acid will form, so it will be acidic in nature also, more lactic acidosis will be there. Because cancer cells have to divide very fast, so they need more DNA synthesis, more RNA synthesis, and so the cancer cells will have high DNA RNA replication process. Not only that one, they have also increased ribonucleotide reductase. Ribonucleotide reductase is a enzyme which normally convert ribose nucleic acid to the deoxyribose forms. So because DNA will be synthesized more, so the activity of ribonucleotide reductase will also increases. And in case of cancer cells, mutated proteins amount will increase and surface area will be damaged. I will show you some of the pictures where it is going to happen. Apoptosis is a normal programmed cells. So normal cells die because of the apoptosis, through the apoptosis. Cancer cells does not induce apoptosis. Apoptosis, what is the process? It is orchestrated with perfect coordination with the pro-apoptotic and anti-apoptotic proteins. In our cells, we are having two types of proteins. We are going to call it pro, which normally initiate apoptosis, and second one, another is called as anti-apoptotic proteins. Anti means which protect us from the apoptosis. So normally in a healthy cells, there will be a balance between pro and anti-apoptotic proteins. In case of, like, some of the anti-apoptotic proteins are BCL2, BCLXLs, these are anti-apoptotic, and pro-apoptotic proteins are BAX protein, BAC protein, BCL proteins. In case of cancer cells, they are having high amount of anti-apoptotic proteins. So they are having more amount of those proteins which normally oppose anti-apoptosis. So high amount of anti-apoptotic proteins, whereas the apoptotic initiacy protein, that is pro-apoptotic protein, that is less. So they have less amount of BAX, BCL and other proteins, whereas they are having more amount of BCL proteins. BCL2, BCLXL, these proteins will be in high amount. Cancer cells depends normally on the glycolysis for their energy productions. So they do more glycolysis and in the anaerobic fashions also. So this required high metabolic requirement for the cancer cell, because they have to divide more, so they require more energy. So because of that one, they are having more glucose requirement and more oxygen requirement. To fulfill these two requirements, they normally have high angiogenesis. So if you see these pictures, this is a normal cells which is having a regular cell surface, and they are going to die with the apoptosis. Whereas in case of cancer cells, they don't have a regular shapes, they are having normally pseudopodia like a structure on the plasma membrane, and they don't go for death, they don't have apoptosis. This is again normal cell surface, which you can see regular surface and the light color of nucleus. Whereas in case of cancer cells, cells grow on multi-layer plus irregular shape. So because of cytoskeleton loss, they will have irregular shapes, plus the nucleus will be of intense dark color because more DNA will be there. So in case of normal cells, more cytoplasm will be there, single nucleus, single nucleolus and fine chromatin. Whereas in case of cancer cells, we'll have more DNA, so cytoplasm reduces, multiple nucleus because more DNA will be duplicated, and coarse chromatin will be there. So these are the basic differences between the normal cell and cancer cells. These things already we have discussed in the details. This is just summary. Cancer cells will have irregular shapes, normal cells will have a regular shape, regular surface, regular plasma membrane. Nucleus larger and darker because of more DNA synthesis in case of cancer cells, whereas normal cells will have normal 1:6 ratio will be there. Growth, here it is out of control, unobedient cell, more DNA replication will, more cell division will be there. Whereas in case of normal cell, it is systematic, regulated fashion, and it is under the control. Cancer cells are immature and normally not going to die if it not having proper apoptosis, whereas in case of normal cells, they are going to die with the apoptosis. Now cancer cells lost the normal communication processes, they are not responsive to the any kind of response, any kind of signaling systems. Normal cells going to respond to the communications. It is cancer cells normally adopt some strategy by which they hide themselves from our immune systems, so they are invisible to the immune systems, whereas normal cells going to be have visibility with the immune system, they are going to be visible. More angiogenesis in case of cancer cells, angiogenesis only when we requires it. Cancer cells requires more oxygen, this requires oxygen in the normal manner. Glucose, cancer cells require more, normal cells require some amount of glucose according to their requirement. Environment, because of more lactic acid formations, cancer cells environment will be acidic, whereas normal cells environment will be alkaline. And the nutrient which cancer cells prefer is glucose because it is easily metabolized, easily it gives energy, whereas normal cells can use fat also, can use ketone also, can use glucose also. So these are the basic differences between the basic characteristic feature of cancer cells, which is different from the normal cells. So we have seen today the difference between the benign tumors, difference between the malignant tumors, what are the different characteristic features, and then we have also differentiated between the normal cells and the cancer cells. So this is all about the today. Next day we will talk about the etiological factor or the molecular basis of the cancers. So thank you very much. If you have not subscribed my channel, please subscribe it. If you like the video, press the like button. So thank you. Have a nice day.